I just recently finished my PhD research at the University of Alberta, and so what I'm going to be talking to you today about is that research and some of the experiments I did during that time. I'm using two screens, so I apologize if there's a bit of lag here. I'm just going to get used to that. Okay. So I like to get the acknowledgements out of the way to begin with. There were a lot of people who helped with this project, and without them, it wouldn't have been possible, but I'd particularly like to acknowledge my two supervisors, Dr. Ann Nath at the University of Alberta in the Department of Renewable Resources, and then Dr. Shelley Proust, who is a species at risk biologist with Parks Canada. She was my co-supervisor. And then this research was completed in Grasslands National Park, so they supported us in a number of ways with funding, logistical stuff with research, and I just want to acknowledge them as well. But without further delay, let me introduce you to the greater sage-grouse. So this is big and beautiful bird. It's about, it's a lot larger than a chicken, so it's about that size. It's an iconic prairie species, and it's best known for its unique mating ritual. So this guy, the males, as you can see in the top left-hand corner there, they have these air sacs on their chest, and every spring the males gather together at an area called a lek. It's the same area every year. They really have a high fidelity to those sites, and they come back and they gather and they dance and they strut and they pop these air sacs on their chest to try to get the attention of females. The females are really picky, and about 98% of them will choose the same 1 to 3 males to reproduce with. And it all comes down to that dance. So that's what they're known for. And when they pop the air sacs on their chest, it makes this sound that's like popping a cork of champagne, and that sound can travel up to 3 kilometers away from that lekking ground, and that signals the females to come in and look at the dancing. So as I said, the grouse will return to the same spot every year. So this is essentially the center point of where their habitat expands from. So you have this central point of the lek, and then once the mating is finished, the females will disperse from there to find nesting habitat. They go on average about 3 kilometers from that point to find good nesting habitat. And from nesting habitat, when the chicks hatch, they disperse from there a little bit farther out to find good areas for brood rearing. And in the winter, they disperse a little bit farther for wintering habitat. But essentially that lek is a center point, and any kind of restoration we might want to do for this bird, we'd have to know where that lek is and place the habitat appropriately based on the needs of the grouse throughout the year. Now, unfortunately, the sage-grouse populations have been declining by really drastically. 98% declines in populations since 1968 in Alberta and since 1988 in Saskatchewan. So right now, and this number is current as of 2018, we have between 300 and 350 individuals left in Canada. Those birds are distributed throughout Alberta and Saskatchewan. And there are 2 leks, so 2 populations left in Saskatchewan, and they're both within Grasslands National Park. Now, historic estimates for populations place the grouse at millions. It's a rough estimate, of it's been about, I think, 1 million to 16 million is the range that they put the historical estimates in before European settlement. And now, throughout North America, the populations in Canada and the US, we are at tens of thousands. So declines have been dramatic through Canada and the US, and consequently, we have in Canada listed the greater sage-grouse as endangered with the Species at Risk Act, the federal act. Before that, COSEWIC recommended the listing independent of SARA, and then the provinces have also listed the species as endangered. And I think that makes sense. And with this listing, you have additional protections for the bird. You have not just protection of the bird itself from hunting and harassment, but you have protection of the habitat. And you also then have a recovery strategy drafted up to figure out how we can best try to increase populations. So the main reason, though, that these population declines have occurred was initially because Europeans came in and there was a huge loss of habitat, fragmentation of that habitat, and degradation. And so that's the reason those populations have gone down. We in Canada are looking at 7% of historical habitat remaining for the great sage grouse. And if you look at this map, you can see the dark green is the remaining habitat at the northern tips in Canada, just in the southern parts of Alberta and Saskatchewan, and that's where Grasslands National Park is situated. And even though we have 7% of habitat left, a lot of that remaining habitat is actually suboptimal for what the species need to thrive. And so we're continuing to see these population declines just because even though it's undisturbed, potentially habitat, it's not what it needs to be. So not only are we looking at trying to restore habitat in areas where there isn't any, we're looking at trying to enhance the habitat that does remain because it isn't really meeting the requirements of the grouse at this time. Now, suboptimal habitat could look like a number of things. It could mean that native prairie contains now human structures, which then become exploited by predators, which will, drive populations of grouse down further. Corvids are big on that. They will sit on top of fence structures or high poles, anything above 2 meters, and they're able to predate the nests effectively that way. Fragmentation is a big thing. Roads cause mortality just hitting the grouse. And although they're very beautiful, sage grouse, they are not described as the smartest bird. So fences are particularly problematic for them. They have a high mortality rate with just collisions to fence lines. And you think they'd be able to see it, but they don't. And so this is also a problem. That's what degradation of the habitat can look like. Grouse require specific vegetation cover throughout the year. So for example, forbs, flowering species, are really important for hen and chick nutrition throughout the summer. And if we have some forbs, but they're below the sort of required level, you're going to see a lot more chick mortality throughout the summer. And again, that will drive populations down. But the biggest thing grouse need is sagebrush. So as their name implies, sage grouse are sagebrush obligates, which means they require sagebrush to live. And grouse have very specific sagebrush requirements for lekking, for nesting, for brood rearing, and for overwintering. So 4 different habitat types for this bird. And each of those habitat types have different sagebrush cover requirements. So sagebrush is so important because it provides cover from predators. The grouse nest almost exclusively under these plants. There are other shrubs throughout the prairie ecosystem, but they will not nest under any similar kind of like shrub with the same morphological characteristics, always sagebrush. And in winter, it comprises 100% of their diet. And that's because these shrubs are one of the only vegetation species in this area that retain their leaves over winter, and they're tall, so they sit above that snow line, so the grouse access that during the winter and eat almost exclusively sagebrush leaves. So if this plant is not there in the required amount, it can have really major consequences on the grouse population. In the Canadian range of the sage grouse, we have a specific species of sagebrush, just one, and it's called Artemisia canna, or silver sagebrush. So it's the dominant sagebrush, and it has a little bit different morphology compared to Artemisia tridentata, which is called big sagebrush, and it is highly present, very dominant throughout the US. And a lot of the scientific literature has been conducted on that species. But here up in the northern range of sage grouse habitat, we don't have that species. So restoration has been really difficult with Artemisia canna because we just don't have the science to back anything up. And unfortunately, the ecology of these two plants is very different. So trying to apply restoration techniques from big sagebrush onto silver sagebrush, it doesn't really work. So silver sagebrush, it's quite unique because it reproduces both sexually and asexually. So it can produce thousands of seeds from one individual every fall. But the majority of reproduction is done through underground rhizomes. And so this is a really neat strategy for the plant because fire is a dominant disturbance in the prairie ecosystems. And because it has these rhizome systems that can resprout, it's really resistant to fire because it can resprout from those rhizomes afterwards, as long as the fire severity isn't too great. It's also capable from resprouting from the crown, which is good. It makes it a little bit more tolerant of disturbances. But we've had a really difficult time trying to restore this plant from seed and from seedlings. It's just revegetation has been very difficult. So in restoration, we have about 5 to 6% success rate from broadcast seeding and only 25% survival rate from after 5 years from planting seedlings. And when you think about the amount of effort that goes into collecting seed and growing it out and then getting all the volunteers and planting and, walking these hectares with broadcast seeding, it's just not feasible to have successful restoration if we're remaining at this success level for revegetation. So we need to increase that so we can actually do habitat restoration effectively. The other thing is we really don't know much about this plant specifically. We don't know at what age sagebrush reaches its maximum height or canopy cover. And that is important because the individual canopy cover contributes to landscape cover. And if we don't know how long it's going to take to get that maximum canopy cover, we can't make an estimate of how long it might take after planting to reach those habitat targets we're trying to achieve. So that's also something we don't know and something that my research was trying to address. So with all of that information, my research for my PhD really aimed at developing methods to improve the establishment, growth, and survival of silver sagebrush because it is a critical component of greater sage-grouse habitat. And habitat restoration has been cited in the recovery strategy for the grouse as a way to increase their population. So if we have better habitat and more of it, the grouse populations should increase. Now in Grasslands National Park, we are sitting at about 10% sagebrush cover maximum in only specific places. So this also is really critical for that area and those populations specifically. But these techniques that I've developed, I hope can be also used throughout North America, not just in the park and throughout Alberta, because Artemisia canna is distributed throughout the US and it's a critical part of that sagebrush system, but it's just not the dominant species. And so what I did to try and meet this objective is 4 different experiments. The first was looking at sagebrush ecology, and that was to develop planting or seeding densities to obtain desired cover. And then we have 2 experiments that were looking at improving that seeding and container growth outcomes. And experiment 4 was running all of that sort of revegetation stuff through the filter of land management. So when we actually then do this and apply it to the field, what does the past land management, current land management then do to alter the restoration outcomes? And hopefully when we put that all together, we're looking at, a best management practices for sage-grouse habitat restoration. All these experiments were independent of each other. They didn't really build on each other, but they definitely fit together like puzzle pieces and provided critical component of the overall restoration picture for this to be successful. Now today, because we have limited time, I'm not going to be talking about experiment 2 or experiment 4. But if you do want to ask me anything about that, I'd be happy to talk about it after. But I'm going to be focusing today on experiment 1 and 3. So experiment 1 was looking at modeling Artemisia canna cover as a function of plant canopy cover, density, and age under different grazing regimes. And again, I was doing that to develop these models that we can find planting densities and seeding densities so we can reach those habitat cover targets during restoration. So, and why we need this is because habitat targets are expressed in percent cover. So for example, 30% sagebrush cover. However, in restoration, we work in densities. So we use kilogram of seed per hectare or number of seedlings per hectare planted. And what we don't currently have is a way to determine how many individuals to seed or plant to obtain that desired cover. So my objective for this experiment was to develop a model to accurately predict cover of Artemisia canna from planting or seeding densities at any given time post-restoration. And that broke down into first assessing how well morphological traits like stem diameter could estimate sagebrush age, and second, relating those morphological traits to sagebrush density and cover on the landscape. So what I did is I measured naturally occurring sagebrush stands in Grasslands National Park. I divided the park into 3 distinct ecological areas whose grazing histories were also different. And there were 2 cattle grazing areas, West Block, where grazing occurred at greater intensities, and East Block, where grazing was lighter. And there was 1 actual bison grazed area in West Block, which was pretty cool to be working in a bison area. So I measured about 15 plots in those areas and ensured a range of sagebrush densities were also measured. So I designed or designated a 10 by 10 meter area around a selected sagebrush stand, and that's sort of indicated in the picture here on the right. And cover was measured using the line intercept method, which is shown in the diagram. Density was simply the number of individuals whose stems were within the plot. And then on each sagebrush, I measured crown diameter, height, and the diameter of the largest above ground stem. And on a subset of individuals, I also measured canopy cover through photo analysis and age via ring count. So photos were taken as centered as possible from above and processed with ImageJ software to determine canopy cover in centimeters squared. And that's depicted here in the photo. So I took a photo, made it binary after I isolated the canopy cover, and then used the scale bar to essentially look at the cover that was resulting from that photo. And then to determine age, I took a cross section of the root or stem, and then this was sanded to bring out the rings. And then the rings were counted on high resolution scans of those sagebrush stems. So this is what that process looked like. It was kind of quite labor intensive. And of course, because we had to destructively sample these individuals, we only sampled 5% of all of those, or 4% of all of those that we actually measured. So we were looking at taking about 150 individuals total for this experiment over a really large area. Okay. This part is just about statistical analysis, but essentially I used a Curve Expert software to fit multiple different models to the morphological characteristics. And then I used different parameters to assess best fit, like AIC, residual distribution. I used an ANCOVA to determine if relationships between those morphological traits were different between the grazing areas. And then I used age demographics from each sagebrush stand to determine how sagebrush density changed over time. And that was interesting because ideally we could have had a long-term restoration plot set up where I could monitor someone, not me, because it would take years and years and years, but multiple someones could monitor sagebrush age as an individual actually grew over a 50-year time, but we don't have the luxury of that time period. So I did the best I could with using age demographics to look at mortality that way. And then I basically modeled landscape cover as a product of that canopy cover of an individual multiplied by the density. And the density changed using that age demographic curve. So what I found was that there were really strong morphological relationships between certain aspects. So there was a significant linear relationship between sagebrush age and stem diameter. And that allowed me essentially to estimate age of individuals that I didn't want to destructively sample. So I could just measure stem diameter and have a pretty accurate estimate of how old that individual was, which then allowed me to use 3,000 different data points in this, the rest of the models. And this was pretty cool because our research showed, just from the ring counts itself, that these individuals are living a lot longer than previously thought, up to 44 years with the actual ring count and 94 years with the actual linear equation here. And why it's so much greater, 94 years using the linear equation, I didn't want to sample any of those really, really large individuals, a lot of them, because they contribute a lot of cover to the landscape, and I thought they could be quite important to the reproduction of those plants going forward and for the habitat. So I didn't want to actually remove those from the landscape. What I also saw was canopy cover was significantly related to age and also stem diameter, which was a good thing, I thought, because age and stem diameter were related. And basically, those relationships could be modeled as reciprocal quadratic equations. The R-squared value was pretty okay for me to accept that. And then this was interesting because the peaks are indicating that maximum canopy cover is similar for all of these areas, but that the time at which these plants obtain the maximum canopy cover was quite different. So if we just look at the bison grazed area, those peaks are a lot further over than, say, in the cattle west area. So it's indicating growth is slower in those bison grazed area for some reason. Looking at the age demographics, we found like a high degree of individuals large proportion of individuals are between 1 and 3 years. So we're seeing a high mortality at the beginning of life, and then few individuals remain on the landscape after that. And I use this to basically come up with a sliding scale of density over time. And I used a logistic model for that. So when I incorporated that equation of density into the landscape cover equation I have here, so density over time multiplied by the average canopy cover, I had these models for landscape cover on the cover of sagebrush on the landscape. So what was immediately apparent after putting this all together is that even at relatively high planting densities of, say, 1 plant per meter squared, which is indicated by those dashed lines, we have really low sagebrush cover outputs or predictions. And so this high mortality and the slow growth rate of the sagebrush is really limiting what we could achieve with restoration, even at high planting densities, is what this is telling us. When I compared those predicted values that from my models there with the measurements I made in the field, all the predictions are within 10% of the field value. And in the cattle grazed area, all predictions were within 3% of field values. So this essentially let us determine the planting density we need to obtain specific sagebrush cover in a given time frame. And we didn't have that before. So now, if someone comes in and we say, well, we want to restore 5% sagebrush cover and we want to do it in 5 years, we can determine a planting density to do that, is essentially what this experiment allows us to do. So the models were, I think, sufficiently accurate to target tight ranges of sagebrush habitat. And this is important because the sage grouse need different ranges of habitat in 4 different life stages throughout the year. And the cover is all very different, and they have them need them in specific locations. So it's really important we are able to target those. And but the other thing, like I just said, the high mortality and the slow canopy development really limit what we can achieve with standard restoration revegetation methods. So so that led into experiment 3, essentially, is looking at how can we then really overcome that high mortality rate and potentially increase canopy cover growth or increase the rate at which we obtain that canopy cover. And that's what I was looking at in experiment 3, using nutrient loading as a method to promote those morphological qualities and survival of outplanted sagebrush seedlings. So why I did this is because numerous studies report survival of outplanted sagebrush seedlings between 25 and 36% after 3 to 5 years. So that's quite low. And it's extremely low when you think about the resources and time you need to develop and plant those seedlings. But some studies on trees have shown that increased nutrient content in tissues and increased competitive ability, which is essentially the size of the plant, confers an advantage for survival and growth after planting. And other studies have done this by manipulating the nutrient availability in the greenhouse, and that could potentially nutrient load seedlings to give them these advantages. Nutrient loading is a fertilization regime that's applied in the greenhouse, and it increases plant nutrient reserves. So continuing to fertilize seedlings when biomass accumulation has plateaued or slowed allows nutrient content in tissues to increase. And we can really optimize that here before we see any toxicity symptoms in the plant. So that's kind of like where we want to work when we're thinking about nutrient loading. So the objective of this study was to apply the nutrient loading concept to Artemisia canna in 2 phases. I mean, this has never been done on a shrub before. It has been done on tree species, but this is relatively new for shrubs, especially in a grassland region. So I didn't know if it was going to work at all. So the first phase was looking at figuring out if it would work, and working in the greenhouse to identify a fertilization rate and development time required to produce nutrient loading seedlings. And then the second phase, looking at whether or not those nutrient loading seedlings actually do any better in the field versus their conventionally grown fellows. And so in phase 1, I grew seedlings for 12 weeks in the greenhouse, and each seedling received 70 milligrams of nitrogen during those 12 weeks under 4 different fertilization schedules. These are depicted in the graph here, where we have a single dose or a constant dose of the same amount every week. And then we have these exponential and modified exponential fertilization schedules. And those are supposedly going to be the ones that result in nutrient loading, because they mirror the biomass accumulation of the plant and the actual match the nutrient demands of those plants as it's growing. So this is what I looked at, 4 different fertilization schedules, but they all received the same amount of nutrients. And throughout those 12 weeks, every 2 weeks, I assessed seedling health on a 1 to 5 scale, indicated kind of at the top, in the top photos. I looked at root and leaf mass, leaf area, root and leaf nitrogen. And essentially what I found is that nutrient loading was not successful, largely due to the fact that growth did not plateau during that 12-week period of greenhouse growth. And I think this is a really important finding, because standard greenhouse development of these sagebrush seedlings, they're only grown for about 12 weeks. They put them in mid-January after Christmas, and then we're planting them in the field at the beginning of May. And so, you know, you've got about 12 to 16 weeks to play around with how much you can put in these plants, and we're not seeing growth plateauing. So they're not really able to accumulate nitrogen to a sufficient level for nutrient loading. And that's indicated here in the graph with leaf mass, showing it's continually increasing. And then if you look at the graph to the right, we're looking at the leaf nitrogen content. You can see it's slowly decreasing. Actually, it's doing the opposite of what we wanted over those first 12 weeks. So what I did was increase growth time for phase 2, and I also increased the amount of nutrients the plants were getting, because it seemed like they were not receiving enough. So in phase 2, I grew the seedlings for 26 weeks. I experimented with different amounts of fertilizer this time, not just using 70, but more. But the same 4 schedules for fertilizer administration. Before they went in the field, I looked at the nitrogen content of the leaves. I also looked at that after they were in the field for a certain amount of time. And when they went and transplanted into the field, I monitored their health, height, and canopy cover. This is what our field plot looks like. So this was established in Grasslands National Park. So the seedlings were exposed to the same field conditions they would be as if I had planted them into potential sage-grouse habitat. But I did remove the competition from around those plants because I really wanted to isolate the effects of nutrient loading. And there were other studies that looked at the effect of competition on sagebrush seedling health and survival. So I used that to infer what that might have been if we didn't remove the competition. So basically for the stats, I was using ANOVA. I also used a linear mixed effects model to determine if the position in that plot had affected the seedling growth. And I used a generalized linear model to look at survival with a binomial distribution. And what I found, which is really exciting, is that actually nutrient loading worked this time. So it seems that 26 weeks was an adequate amount of time for these seedlings to grow in the greenhouse, for biomass to slow, for them to slowly accumulate nitrogen in their tissues. And you can see that in the graph here, you have leaf nitrogen content on the y-axis and then the treatments along the bottom. The green bars indicate the exponential fertilization schedule and the brown are the modified. So those groups are a little bit higher than the conventionally grown seedlings. And at the higher nutrient amount that they were given, those bars are significantly greater than some of the other treatments that received less nutrients. What was interesting is that nitrogen was stored in a 1-to-1 ratio in leaves and roots, which just kind of indicates the different strategies this plant has for drought tolerance and things like that. And we did see a trade-off between nitrogen and carbon, which was, or carbohydrates, which was expected. So that was good. So, but the big question was, did the nutrient loading seedlings actually do any better when we put them in the field? And I'm happy to report they did. So we found that the nutrient loaded seedlings had an increased survival of about 80%. And that unloaded seedlings had a 50% survival rate. And this is after over 2 years. So that's not just in a first year, it's after 2 years. They're still maintaining a pretty good survival. And when you compare that to, 36% over 3 years, I'm really, really happy with that outcome. But not only that, and this was a huge surprise to me, canopy cover of the loaded seedlings was almost double that of the unloaded seedlings. And this was so unexpected. And you can see this amazing explosion of growth here in the photo. This is the same sagebrush plant. It's the photo I took to analyze cover at 2, 5, 14, and 15 months. And you can see between 14 and 15 months, this plant just doubled in size. It went from about 20 centimeters across to 40. And it was just amazing to come back into the field and see that. Okay. The other really, really cool thing I found in this experiment was that these plants were capable of reproduction almost immediately. And that was interesting because the other, previous literature is showing, we're estimating that reproduction really doesn't start until 3 years on. And I mean, we were seeing some of these sprouts developing in the greenhouse. This is a photo from 25 weeks. After greenhouse growth, you can see it's already trying to re-sprout from the root ball, which is very, very cool. So we returned to the field after 4 months, and we had 6 new individuals in the plot. At the end of the first year, we had 7 new individuals in the plot. Then there was no way that that was coming from any of the native ecosystem around. There was no sagebrush in that area, so that was very, very exciting. The other thing, though, that this study indicated to us is that the microsite selection, or where you plant sagebrush, is going to be very important. So that the analysis of the data was showing that position in the plot had some effect on the growth of those individuals. So selecting areas where you have better soil conditions is definitely important for their growth and survival. What I concluded from this is that we need to extend the growth period for sagebrush seedlings, especially for these restorations in Grasslands National Park. Talked to the greenhouses who develop seedlings for Grasslands National Park, and they're totally on board, so that's great. We are kind of implementing that. And the other big thing is if we do nutrient load seedlings, we can expect increased survival and canopy development, meaning accelerated restoration timelines and potentially less replanting or management farther down the road to achieve those habitat covers. So that's a really awesome result of this. Bringing it all together though, because together that's where it gets really, really interesting. Remember in experiment 1, I modeled sagebrush cover and concluded that increases in cover would be capped at about 3% at high planting densities, and this was due to slow canopy development and high mortality. Well, in experiment 3, I found a way to overcome the high mortality and increase the canopy cover development. So when we put those results together, basically incorporating those new canopy covers and mortality in those models I developed, we could increase cover predictions by a lot. So what we actually see is increases of 42% in bison grazed area, 136% in cattle grazed area, in East Block, 38% in cattle grazed area of West Block. So these increases in cover due to experiment 3, that nutrient loading really put successful restoration of sage-grouse habitat within reach. So now we have the means to do that, and we know how many to plant, where to plant them, when we're going to get the expected habitat cover targets. So that's kind of what came out of these 2 experiments for my PhD thesis. And yeah, just to summarize it for you guys, over time, basically the results help overcome those impediments in restoring sagebrush by providing important information about sagebrush growth and their persistence over time to inform seeding and planting rates, and then overcoming exceedingly high first-year mortality of outplanted seedlings through nutrient loading. And I believe that is the end of things.